Hi there. My guest this week is Sean Sullivan, a biotechnology PhD candidate studying at Tufts University in the Boston area. Sean is a bright young mind eager to advance his knowledge of synthetic biology after spending some time in the private biotech industry, honing in on his true desire to bring positive change to the world. In particular, Sean is studying how to engineer the common yeast, Saccharomyces cerevisiae, to biosynthesize alternate forms of carbon as food for efficient and environmentally sustainable synthesis of valuable compounds such as fuels, medicines, and more. On our chat, we covered a variety of topics that range from learning how the PhD process works, to how he funds his education, the role of formal education versus those who go the DIYBio self-taught route, the research his lab is doing into the gut microbiome, and our shared passion for the potential of genetic engineering. I'd like to thank Sean for his time in Tufts University for allowing us to take a peek into the lab where Sean and his fellow academics perform their research. I really enjoyed this conversation, and I think you're going to enjoy it too.
For folks who don't know you, maybe you could tell us a little bit about who you are and what you're up to these days.
Hi, my name is Sean Sullivan, and I'm currently doing a PhD in Biotechnology Engineering at Tufts University, entering my fourth year. So I'm from Massachusetts, so I haven't gone that far. Was always interested in biology and sort of like why life, like why does life exist? Why aren't we just like the rocks next to us? How are we different? And in college, so I guess this is like the whole journey of getting to where I am now, if you don't mind.
That's please.
The short of like synopsis. So in college, I was interested in life, so I studied biology, and I had an awesome teacher who taught me microbiology in Frank Fekete. So I started doing research in his lab a little bit as an undergrad and was just so blown away by what microbes, which we think of as these like incredibly simple organisms, are able to accomplish, and all the different sort of solutions that they've come up with for a lot of the same challenges that humans face. How do we protect ourselves? How do we like get food? How do we, resist attacks by other organisms, survive harsh conditions? So that sort of got me thinking about bioengineering, thinking about like, okay, how could we take these things that microbes can do? And I looked a little bit at biofuels type companies that existed at the time, but this was sort of the tail end of the biofuels craze about 2011 when I was graduating college. And they weren't really taking people. That was kind of around the crash of biofuels. So I ended up joining a molecular diagnostics biotech startup. And I was there for a bunch of years and really, really loved the sort of, wearing many hats, watching the company grow, everybody working together and everybody really knowing each other very well and pulling together to accomplish these goals. But after a few years, I started to feel like I don't really have the knowledge base to be someone that's driving the research that we're trying to do. And still interested in synthetic biology, bioengineering, had been monitoring it the whole time. I was like, okay, I'll go back, do a PhD in this field, get like a strong technical foundation that I could transition to, working at a startup again, or, whether it's like more on the business side, we're trying to understand both like the technical aspects of what people are trying to do and how that would translate, where that would fit in a market. And I've just been doing the PhD now, and that's what I've been grinding on. So that's kind of up to the present day.
Yeah, so you're in your fourth year?
Yep.
How long until you graduate and get that PhD?
It'll probably take 5 years, plus or minus 6 months, I would say. So I'll have basically 2 more years. I think it's very variable in terms of like, it's not a linear process, the PhD. Like you end up trying, okay, get my finger on the screen here. It's not like you just proceed through it in sort of like a nice, easy track. It's sort of like you're somewhere and then you try a few different directions and then all of a sudden something works and then you keep going in that direction and spread out again.
And you mentioned it specifically in biotechnology, or is it in synthetic biology?
Technically, it's biotechnology engineering, which is sort of like, the chemical engineering industry, which is pretty mature at this, at this stage, have a lot of like sort of engineering principles that get applied to these large scale chemical transformation processes. And it's sort of like trying to apply those same processes to producing things with cells. There's a lot of similarities. Like instead of, a big distillation tower, you might have just like a bioreactor. Like, is that a bioreactor? No. But yeah, so those principles, but obviously also like the program, I take a bunch of classes that teach about this type of engineering, but the real meat of what you get into is in your thesis research. So I tell people, when people ask, like I say, I do genetic engineering because my actual day-to-day is engineering yeast and bacteria, genetic engineering. And like the sort of biotechnology applications are like the stuff that comes into play when you start really scaling up.
That's great. I want to get into that too. Maybe you could talk about, because I'm not familiar with it, what does the PhD process look like? So you mentioned that it's not like undergrad where there's 4 years, there's a set curriculum, a set path.
Yeah.
As long as you take the right amount of credits per semester, you walk away with a degree after a certain period of time. There's some variable part of the PhD process.
Oh, yeah.
What does that look like? I mean, why is it variable? And you could talk a little bit about what the process of getting your PhD is like.
Yeah, so this is like could be a whole interview in and of itself, and obviously different programs are different areas are different. So my process was the first year you're basically taking classes and you start to get into lab. You get sorted into a lab in October, but you don't really sort of start ramping up research until late in the spring. You take a qualification process, which is a painful, like proving in front of all of the faculty in the department that you understand the basics of what you've learned in the classes. You present like your proposed research project to them and they comment on it. You have to kind of defend it and justify like why it's important, why this is the right way to do things. And then you start doing research, and the variability comes in, like I said, by program, by the lab that you're in, by the project that you're on, and also just by the area that you're in, like very, very broadly. So like, because I actually work with living organisms that take time to grow, it naturally limits the number, like how quickly I can iterate, the number of experiments that I can run. If something goes wrong or I need to make a pivot, my pivot's going to take me a month, as opposed to, I would say the other one that I think about is people who still work in bioengineering or biomedical engineering, and they do more software engineering based aspects of it, like trying to, bioinformatics, where they can, all the data that they're going to work with. A lot of these labs don't work with any data at all that they generate themselves. They take it from databases. So it's all sitting there and you can just start working. You can set up a program, and even if it's a, like a very computationally intensive thing you're trying to do, schools provide resources that you can run these clusters on, clusters that you can run your code on. You can set it and forget it, walk away and go be like doing reading and thinking about it. Whereas like I have to spend a lot of time in the lab, making all the DNA, doing the transformation, taking care of cells. So I think that's sort of like the actual study area part of it. You can get into a lab that has like a very, very clear foundation and sort of area that they already have a lot of expertise in, and sort of take that ball and run with it. I joined a lab that before I joined, I think just this spring, my PI got tenure. So still kind of a lab that's getting going. There's a lot more exploration that goes into it.
What's a PI?
Oh, principal investigator. So they're the person who sort of is like, it's their lab, and they provide the funding for everything that happens in the lab by writing grants and applying for money, basically from different, either governmental or non-governmental organizations.
Is
Yeah.
that how you're able to fund yourself? Like if the PhD process can take upwards of 4 or 5 years, most people who are getting an education often don't have the time to work at the same time. Like, are you working at the same time? Like, how do you
No,
sustain
this is
yourself?
yeah, so this is a full-time thing, but I actually get paid. I have a stipend, so this is not me paying anything. It's a low amount of money, definitely, which is not something that I'm wild about. But at the same time, like I get paid to do really interesting research, and I've definitely
Absolutely.
been building like the knowledge base and sort of the understanding that I hoped that I was going to be building 3 years ago when I started, which is pretty wild. So it's not a lot of money, and I don't think
Do they cover their cost of education too, or yeah?
yeah. I don't.
So if you
I
get
mean,
accepted into, is that pretty common? Like if you get accepted into a PhD program, most of the time the education is free and you get a stipend, or is that unusual, or is that limited to
I'm
just?
not a grad student expert, but it definitely varies by field. So I know that like it's all about the money. So I know that for like our department, we have a good amount of money because we have a good amount of grant money. So I don't have to do that much and get paid, but I know that like a lot of times it's a breakdown of like if you go to get a master's, they usually make you pay, and if you do a PhD, they usually pay you every year, even while you're just taking classes, with the idea that you're going to be, generating significant research for the university that then gets turned into more grant applications that brings more money in. So, but like I remember going to orientation right before I started, and it was like you have to attend all these seminars, and one of them was like how to teach a class of 150 people. You know what I mean?
Yeah.
And I don't have to do that at all. Like I have to do grading and office hours when I TA for like a few classes over the course of my entire PhD. And so all of that stuff scared me, but I haven't had to really deal with that at all. The people in like lower, I would say, this is how I think because I'm not an expert,
Hmm.
things like English or, maybe if you were doing like a language study type of thing, yeah, those graduate students, I believe, are expected to do a lot more in terms of teaching duties and like pulling their weight basically in terms of helping teach undergrads. So we have it pretty good. It depends on the area.
Is there a cohort of people that are sort of in alignment with you in terms of your goals and when you plan to get your PhD, or is everyone sort of independent on in their journey?
Well, there's a cohort in terms of when everybody comes in, like, a group of people came in the same year as me, and we all sort of keep in touch and talk to each other. But it's not that unified in terms of like the amount of time that it's going to take, what people are even trying to do. And that's mostly determined by like what lab you're in and what type of research you're doing, which goes back to like some labs can go a lot faster and some labs are like even slower than ours. People that work with mammalian cells, we don't really do anything with mammalian cells. They take a longer time to culture, can be more finicky. You're not more wildly so, like a lot of people do it, but those experiments can take longer.
Yeah.
Like if you're doing anything in mice, you have to, years out,
Mm.
let the school know that you want to do mice experiments. They have to make sure that you have the facilities for it. So there's things that can, and you have to like, if you need a certain genotype of mice, you have to make sure you raise it, or get it from somewhere. You have to like maintain these things, and it's just gets messy.
Yeah, yeah, that's really interesting. So how many people are in your lab also working on
Right
GMO?
now, it's about 6 that are in lab. We just graduated a few people over the past bunch of months. We were up at 10 people about 6
Oh,
months
okay.
ago. So,
Okay, I
um,
would say, I don't know if that's typical or not, but it's a pretty small group of people.
so it's not huge. It's a good amount of like when we were at 10 people, that's actually a pretty good amount of graduate students. Um, it's like a small lab compared to some like really, really large labs, but I think it runs the whole gamut where some people, they will literally only take undergrads to do research, in which case you're getting like 1 or 2 years. Things move pretty slowly. And some of the really like diesel famous labs also don't even have that many grad students and are predominantly postdocs, people that are there that have already had their PhD, just like cranking away on things.
What would you say, do you have any insight into the backgrounds, the profile of the other grad students that are in the lab with you? Do they kind of fit your similar background where they did undergrad, maybe they went into the private sector for a little bit and then they came back? Do you have any like adult learners that have had a long span between their undergrad and wanted to change careers? Do you have any insight into that?
Yeah, I mean, I can speak to the people in my lab and say that there's some variety. I think everybody, no crazy stories in terms of somebody coming from like an English undergrad major, but definitely some people that went straight from undergrad, knew exactly that they wanted to get a PhD immediately, and a bunch of people that did, you know, worked at a private company or spent a couple of years doing something else. Haven't seen any like, you know, people dropping back in at past age 40 or anything like that, lifelong learners. But I don't think that that would be frowned on at all. I think that's...
Do you find the type of research that you guys are doing in the in grad school, is that really, so I'll just share a little story. A lot of times in the amateur mushroom growing community, people, especially young people, they get excited about growing mushrooms at home, and they decide when they're in high school that they want to go and get a degree in mycology because they want to study fungi and they really want to go deep there. But what they discover is that there's not a lot of undergrad programs in mycology. It's typically a grad degree type program. Do you kind of feel the same way about the type of work that you're doing? Is that level of work, biological engineering, only accessible at the grad degree, and how do you feel about that? Should more of that be available to people in undergrad?
Well, I think for the mycology experience in particular, I think it's not that it's not available. I mean, I guess it's not available, but it's kind of a specialized thing, you know, to start focusing just on one organism. Whereas in college, usually you're taking classes around just like molecular biology in general, genetics in general, sort of like the broader concepts. So in terms of diving into one particular thing, I kind of understand why that a lot of times waits for graduate school. But I don't see, I really like the idea of, you know, the DIYBio community being able to provide an outlet and maybe a knowledge base and, you know, like we were talking about before we got on the call, things to think about for maybe building a home lab, how to go about doing that for people that are really motivated to like dive into their own sort of specialization that they want to get into.
Yeah, I wonder if you could talk a little bit about that, because in the last podcast we talked to Dakota, which is pretty much self-taught. He did go to undergrad, but he didn't get a grad degree. I think he did. He did say he dropped out of high school, and here he is. He's a scientist with a biotech startup in Boston, and quite successful at it, I'd say. What is the sort of and I'm of the belief that there's a place for formal education and there's a place for self-taught learners, and even the DIYBio space, there's definitely a lot of interesting things that the community has done to facilitate the education and learning process. Maybe you could talk a little bit about some of the roles of each of those and the trade-offs associated with them, especially as someone who's like on the other end, you're going all in on the PhD.
Yeah.
And maybe you could just talk about that, because you seem to be encouraging and you like that there's a DIYBio movement. You like that people have home labs. But where does that fit in with people like yourself who are going all in on the education route, the formal education route?
Yeah, well, I mean, this is like, I really liked the interview with Dakota, by the way, and I was really impressed to see that he had like taught himself so much, because and this gets a little personal in that, like, I feel like I've up until relatively recently been someone that feels like you need to follow, like, you know, the path, the normal path. I don't know if I would have had the confidence to do what he did, which is just like, yeah, we're starting a company.
And I think he struggles
We're
with
just
that,
gonna,
by the way. And it's, so I'm fascinated by the fact that he didn't go through this traditional path, and he
yeah,
struggles with it. And I could tell that he wrestles with, like, maybe I should have. And here you are, you're going through this sort of, traditional path, but you're like, maybe I should have started earlier, maybe I didn't need it. So I am
yeah.
fascinated by that. There is no, it just tells me that there's no 100% surefire way to do it.
There's definitely pros and cons to both. I will say that maybe the big contrast, and I think I'll draw a contrast, maybe compare and contrast like software engineering with bioengineering. Especially since, bioengineering and synthetic biology, a lot of like the really key players are very specifically motivated by that analogy, trying to make biology more predictable, more layers of abstraction. I've taught myself some coding and found the online resources to be absolutely incredible. Like, I will type in some question into Google, and I think it's the most like outrageously specific small thing that I'm Googling, and literally somebody has answered that exact question
And probably
online.
100 different ways.
Yeah, exactly. And there's just a whole community on places like Stack Overflow of people who literally they get paid nothing and they will answer your questions and do a good job. It's an incredible community, and there are incredible resources. I think that bio is behind on that, but I think that there are a lot of active efforts to try and build those type of communities, have those type of resources. Like ResearchGate is one. I don't know if you've ever come across this,
Yeah.
because that's the thing a lot of times that pops up when you
Papers
Google
and
something.
literature. Sure.
So papers and literature, but ResearchGate is that more sort of Stack Overflow, like you ask a question and some people will, just post their answers.
Oh,
The
wow.
problem with biology relative to software engineering, sort of the contrast here, is that so many things are just incredibly situation specific, organism specific, gene specific of the particular system that you're looking at. And that can make it harder for just sort of like, whereas on Stack Overflow, you would get, I'm getting this error message. Someone can very easily say, like, somewhere along the line, you change the, the type of object that you're trying to do something to, and it's not happy about it. Whereas with, these bioengineering things, there could be a problem in a lot of different places, or you might just be encountering something that's unique to either your lab, because different, people talk about a reproducibility crisis in a lot of fields, but even things that are like work well, a lot of times, you'll get it into your lab and try it and just not have the same results. And that's not like necessarily anything wrong with the person who published that. Like, things take optimization. You have to figure them out in a way that's more time consuming. And just in general, being in a, having like postdocs in the lab, having my PI, when you run into stuff that seems like it doesn't make a lot of sense, like they just have so much I don't know whether you'd call it like proprietary knowledge or just
Experience.
Just experience, able to answer these questions in a way that it's not, they can cut right to something and just see it immediately in a way that is just very hard to do on some sort of online forum or question thing like that.
Yeah, I would say there's definitely parts of that I agree with. There's things about biology that are non-deterministic, whereas in computer science, a lot of it is zeros and ones. There are a lot of environments and context-specific questions, lots of languages, lots of tools, lots of different ways of doing things, but the reproducibility is much easier, I would say, in computer science than it is in biology, because of just so many environmental factors that play into the results that you might get in the science that you're doing. I'm wondering if maybe we could talk a little bit about your thesis and tell us, if you feel comfortable talking about that. What did you decide to do with your thesis, and what are you researching, and what do you hope to achieve or discover as part of your PhD?
Yeah, so I came in my first year, I was doing a bunch of work engineering the bacteria that live in the gut. so these native gut microbes. Our idea was building genetic tools that would allow them to sense and treat diseases like in situ. So in your gut, they would sense something, and then that signal would be transduced into some response of the cells. And the primary focus of our lab is metabolic disorders like phenylketonuria. So for people that have PKU is the sort of short name for it, the enzyme that breaks down phenylalanine is either non-functional or has greatly reduced function due to a genetic mutation. And so what happens is the phenylalanine is still absorbed, and it actually builds up in the body and becomes toxic. Like, this is something that babies are tested for when they're born, like all babies in the United States, I'm pretty sure. So right now, like, I would say the best treatment that people have is you get daily injections of this enzyme to just replace what your body can't make effectively. And also dietary restriction, where they just have to be very, very careful about what they eat. But then at the end of the first year, so we were going to engineer the bacteria that live in your gut. You would take a probiotic pill of these things. They would just colonize your small intestine. When they sense phenylalanine, they would express, that same enzyme.
So are these genetic, can I interrupt you so I could break that down?
Yeah.
Because even for me, there's a lot that I think could be learned there.
Yeah.
So the probiotic, are these genetically modified bacteria that you're ingesting?
Yes.
All right. So in the lab, someone is genetically modifying these bacteria with the genes that will detect the phenylalanine. I may not be saying that correctly. And then produce that enzyme because the patient's own genetic makeup of their gut biome doesn't produce that enzyme.
Right.
So you're sort of artificially introducing the bacteria that can produce that into the body.
Yeah, and so it would replace the function that normally would be done by your body, and just sort of
you have to
put it back there.
Keep taking the probiotic,
So we're very much sort of designing the genetic tools that would do this, like, sensing and actuation. SynLogic, the company, actually has and one of the people who just graduated from my lab was thinking about working there. They already have like a live engineered therapeutic cell that you take, and you take it. You need to keep taking it. So the goal, I think, would eventually be to have something that's like a much more stable population, but you can increase the therapeutic efficacy without having to do that much work by just taking more of the organism, because it's just a bacteria. It's incredibly easy to make essentially unlimited amounts of this.
Well, here's a question I have is bacteria is its own organism. It's independent of the host, the body. Mm-hmm. How is it that some people produce one kind of bacteria and other people produce a different kind? So,
well, there's not just like a couple kinds. There's massive numbers of different species, and
where do we get when a baby is born? Where does the baby get its gut bacteria from? Do they inherit it from the mother? Or is there something in our genes that encodes these things? How does that work?
So this is not my area of expertise at all, developmental microbiome. But I do know, I read that infants, when you're born, go through 3 phases of microbiome maturation, I think relatively early on. Some of it comes from the mother, some of it comes from diet. Some of the bacteria that we engineer are actually found in breast milk, and that's how they get into infants. Environment, obviously, in terms of what they're eating and what they're exposed to. From the reading I've done about the microbiome, just the house that you're in will have sort of a characteristic microbiome from the people that are living there. And if you moved into somebody's, some family's house, I think it could, your microbiome would start to change over time. So there's a lot of factors that go into it. It's what you're initially exposed to. Diet has a big effect in terms of determining the community that's, what would you call it? The sort of like assembly of the different species that are present. And you know, people think the microbiome is going to be a huge like potential therapeutic area. So they're doing this is like a very active area of research in terms of how microbiome connects to health. And they're starting to learn a bunch of like really interesting things. Like based on your diet, you can sort of feed the healthy bacteria in your gut and help them survive and thrive and take over, you know, be more dominant in the population. Or you can feed, you know, like very simple sugars, and that enables more deleterious bacteria to grow more and take over.
That's really interesting.
Relatively.
Yeah, so a couple of thoughts that came in my mind with that is there's a company, I can't remember the name of them, but they're sort of like a 23andMe for gut bacteria.
Yeah, I think I've seen these ads on Twitter of like, let your genetics tell you, is this the thing? Like let the genetics tell you what you should eat?
Something like that. I think you send them a stool sample and they will do, they'll do DNA analysis of the.
Oh, just the characterization. Yeah.
Which I think is kind of really neat, actually. But
I think it's interesting.
How much you can infer from that, I don't know, but I think it's a neat thing, and I wish I could remember the name of the company.
Yeah, there's a lot that you can infer, but like one of the things that they think so far is, you know, they've found that very different compositions are perfectly healthy. So like two people that are perfectly healthy could have very different compositions. So there's a lot of research in trying to figure out, like people originally thought that there was a healthy microbiome and an unhealthy one. And now they're starting to, you know, see that it seems like there's multiple different configurations that are healthy, and they're trying to figure out why. And my understanding of like the latest thing that people are you know, hypothesis is that different species are able to fill different metabolic roles in the microbiome. And, you know, maybe you don't have the exact same species as someone else, but if all the same functions in your gut are happening normally, you can have a healthy microbiome.
That makes sense. And I like the analogy you use about the house will have different members inside of the house. And a question that came to my mind is the treatment, which is you take a probiotic, which are these engineered organisms that sort of make up for that deficiency. That seems to be one way of addressing an issue with the gut bacteria, but what about modifying the house, right? What about modifying the genetics of the person that's creating the conditions that aren't allowing that enzyme to proliferate?
People are doing that too. I mean, that's, you know, the big promise of things like CRISPR is, you know, that was what excited people so much, was the ability to not just create some DNA construct in a lab and transform it into cells in like a very controlled lab environment, but the potential to potentially edit genes in an already existing person and make that change like after the fact, you know? So I think that's one of the big promises, but that's very hard because you'd have to get this change to basically every cell in the body if you're trying to do like a global change. So a lot of the early applications of this sort of CRISPR of live cell thing, like I know one of the big one is some disease of the eye where I think it's in the retina, but because it's so localized, they literally give you an injection through your eye.
Wow.
And edit the cells at the back of the eye to be able to correct this problem. But it's because it's a much more like limited scope in terms of the number of cells that you need to change. It's sort of like a smaller step to jump onto as opposed to changing every single cell in the body.
Yeah, I wonder if there's a way to like piggyback on the body's immune system, which can have these global responses to proliferate these genetic modifications in the body.
Yeah. Well, the thing is like nothing, there isn't.
Well, I guess if you put it in the blood.
Nothing edits.
Or a virus, maybe.
Right,
maybe a virus that's been, you know, modified to infect you, but infect you with good DNA, not bad DNA.
Right. And that's what a lot of these delivery vectors for these CRISPR systems that are being tested now are, is viral vectors. Yeah, it's tough because like the body delivers stuff everywhere in terms of nutrients, but definitely there's no native mechanism for editing everything. So, this is why it gets into the whole ethical question about like, if you really want to make these just totally systemic body-wide changes, you have to do it at the embryo. Like, you have to do it right at the start, because then that one cell or those few cells go on to make everything. So if you can catch it, right. So it's like,
so is that the main source of contention about doing genetic modification on embryos? Is that it's actually much easier to do it that early than it is.
It's much easier to do it, and that's why it's already been done. Is because like that's the way to make these global changes. But I've actually been really impressed with the ethical sort of caution of the bioengineering community, like the synthetic biology community, sort of recognizing that this is a power, a technology that we could potentially wield without understanding the full consequences of it. And that's why, I don't even remember one, but multiple years ago at this point, there was like people got together and declared a moratorium on editing human embryos. Just basically being like, yes, we can do this. This is really not a good idea to do this until sort of the world has caught up and we would, be able to do this safely and with a lot of buy-in from stakeholders, basically representing like average people. If you've ever seen the movie Gattaca, I think Gattaca does an awesome job of sort of predicting, and that's a movie, I think it's from like the 90s too or something.
Ethan Hawke, right?
Yeah, and Jude Law, and I think Uma Thurman, I think might be in it too.
But they do a dystopian future.
Yeah. But I thought that they did a great job in terms of like coming up with a scenario that's totally believable. Like they do it with genetics, where you get genetic testing, and then you test all the embryos and basically just say, oh, this one's the best. Like that's the kid we're gonna have. And then all of a sudden you have sort of two classes of citizens: ones that have been genetically perfected, screened for all like the best genes of their parents, and ones that are produced by the more just random assortment method and are just basically, not legally necessarily inferior, but like if you're a company hiring or if you're getting life insurance or anything like that, like who are you gonna hire? So I thought they do like a really good job. So there's this whole moratorium, and then people went ahead and did it anyway.
Well, I was going to say, like, it's sort of like nuclear weapons, right? We can have all of these countries agree to not produce them, but there's always going to be these bad actors. And can we afford to not do this research? If nothing else, we have to be able to counter these. We have to be able to counter these bad actors who are, don't necessarily have the same ethical, moral guidelines that we do. I'm trying to wrestle with that philosophy. That's why I like talking about it, because I want to hear other people's perspective.
These in terms of like what the big strategy is, sorts of questions are maybe beyond my pay grade, but I totally believe that it is like I think about it a lot.
I think they're in your pay grade. Actually, I think everybody should be talking about it.
Oh, totally.
You know what I mean?
Well, in terms of like saying like, okay, if we don't do it, then what about China? Like, will China go ahead with it? Like, I don't know geopolitically, but like I very much believe that it's a technology that, like a lot of technologies, nuclear energy, pick one, even like chemical engineering, you come up with like, oh, look at these chemicals that can just kill people. Like, that's stuff that could be used, like chemistry could be used for good or ill. I think it's a technology that has the potential to be used for good or for ill. And I think that you need to be, it can't just be scientists. It has to be multiple stakeholders, and not only scientists and stakeholders, like people that are, experts in ethics. But I feel like there's a gap in terms of communication to the public of like what these technologies are, exactly what they're capable of. You end up seeing, the more fantastic versions of it. Like I think there's the Rock movie where they talk about like genetic engineering making the huge like 10-story tall wolf that he has to fight. Like, that's ridiculous. But, you know, these technologies end up getting talked about by people who their goal is entertainment. I don't think that's necessarily a negative thing, but I do think that there's a gap of like, you know, scientists trying to really build the type of understanding that would lead to really productive conversations.
Maybe
and it's hard. People have a lot of stuff going in their own lives, but yeah.
Yeah, and that's really true with anything really. But yeah, and I also think it's a field that's so technical, and I think it's so far removed from the average person's day-to-day life that isn't a biologist, that doesn't understand how DNA and genetics works. It seems like something that it can only be done at a super high specialized lab. But if you're in the DIYBio space long enough, part of the things that got me excited was realizing the possibility of what you could actually do as an amateur. I could design a piece of DNA and have a company synthesize that DNA, and I could put it in an organism. I don't need to have any special license to do that. There's very little regulation preventing just anyone from really doing that, other than the knowledge of how to do it. And that's incredibly empowering, but also very scary at the same time.
Yeah, and I mean, all the stuff that we've talked about so far in terms of online learning resources and, like, you know, the drastically reduction in cost of sequencing and also DNA synthesis is also the double-edged sword where, like, yes, you're democratizing these ivory tower type disciplines, and you're democratizing it so that, like, even if someone that doesn't have good intentions could be doing this type of thing.
And abstractions, right? Because, like, it's even, I'm noticing with synthetic biology, when you're doing plasma design, it's getting to a level of abstraction that you really can plug and play different genes without really having to dig deep into how they work and the underlying mechanisms that make those genes activate or work. All of those things, I think, contribute towards the power of it and the pace of innovation. But as you said, it's a double-edged sword. So, yeah, it's a very interesting topic, and I really enjoy, like, the philosophical, ethical, conversations about it.
Yeah, I am an optimist, though. Like, I do believe that in general, the people that go into this type of thing are attracted to it, want to do good, and want to build positive things that solve society's problems. I don't know exactly what the mechanism is. Like, I remember when you were talking to Dakota, he said that there was literally an FBI guy that dropped by the DIYBio like hackerspace just to be like,
hey guys,
check for obvious potential problems of someone that's so I think that's good. I
they kicked out a guy that wanted to make a virus, made a comment about we should make a virus and put it on the
that makes me happy that the FBI guy stopped by. But I do believe that most people want to do good with these types of things.
I do too. We derailed a little bit, but I wanted to briefly
the thesis,
recap a little bit about what you're working on and your thesis.
Yeah, so the thesis, so then somebody in my lab published in Nature Communications on engineering yeast. So just basic, like, baker's, brewer's yeast, Saccharomyces cerevisiae. And he, something that is sort of a big goal of genetic engineering biotechnology is, like, making it cost cheaper so that you can start to produce things via biotechnology that are cost competitive with chemical processes, but more particularly, I'm interested in, like, fossil fuel, petrochemical based processes. So, one of the big things that motivates me, and the reason I wanted to get into this field and sort of make it my career, was this idea that this sort of technology has the potential to replace a lot of production of things that are currently produced from fossil fuels and make it much more circular. Whether, so the way that, I don't know if you've talked about this in any of your videos yet, but when you do biotechnology, you're basically having treating some cell as a miniature factory and having it make a molecule or a chemical for you. And so the same way, you know, if you have some huge factory, you need to have inputs, whether it's like metal, heat and energy, or just like the oil itself and derive products out of that. Cells need inputs too, in the form of carbon, energy, nitrogen, all of the different sort of elements that they need to grow and make the thing that you want to make. One of the biggest costs of biotechnology processes is the inputs, like the feedstocks that you have to put in. And for a carbon source, that's mostly just glucose. People, you know, feed it just like very basic glucose, which yeast is very happy to eat and grow on. And even though it doesn't seem like it, glucose at scale is more expensive than other potential sources of carbon and energy. And one of the big ones is biomass, waste biomass. So if you grow corn and you pull up like the corn cob off that plant, you're left with this full plant structure that's actually like stitched together, that the cell walls and this whole structure is actually stitched together out of individual sugar monomers of different types of sugars. So in, you know, the United States, I think I sent you that billion-ton report that the US publishes. Like, the government is very aware of this in trying to sponsor research to figure out, like, how could we turn these, you know, sugars and sources of carbon and energy that we're currently just wasting into feedstocks for biotechnology processes to make that all cheaper and allow us to produce things free of fossil fuels. So our lab, normally what you would do, sort of like the basic, this is metabolic engineering, to get yeast to grow on something that it can't normally grow on, which a lot of these sugars that are constituents of these plants are. You basically give them the genes on a plasmid that allow them to transform the sugar into something that they can metabolize.
I want to take a second and say, like, this, for me, is when I first figured out that this is a potential production mechanism, this idea that you can engineer a yeast, that it's actually not that difficult to even do that. You're going to, you know, design the genes and DNA in the form of a plasmid, literally put it next to a bunch of yeast, and the yeast will take and ingest some of that DNA. I'm very much simplifying it, but it's really not too much more complex than that. No. And just like when you're making beer, you would feed the yeast in a big batch sugar and it produces alcohol as a side product. Well, you can actually engineer yeast to produce all sorts of compounds and chemicals and really valuable things, just like you would brew like beer in a much more efficient way. And that's, to me, that's incredible technology. It's actually where I'm gonna take EverymanBio in its next iteration. But I'm super excited by that because we have an abundance of carbon, obviously, out there.
Right. And like another active area of research is CO2, like capturing CO2 from the atmosphere, because that's just carbon. You would need a different source of energy because CO2 is a highly oxidized form of carbon. But
why yeast? You know, a lot of people ask, well, why yeast as an organism?
So there's a lot of reasons. Some of them are specific to the organism itself. So yeast is a pretty hardy organism overall. It can tolerate high levels of like waste products of metabolism that build up over the course of time during a fermentation and can become toxic, and you basically kill your own cells. They're very good at being resistant to those types of waste metabolites. They are eukaryotes. This is like literally a quiz question that I don't want to go too deep into of all the exact reasons. But
generically speaking, it's a relatively easy organism to modify, right? I mean,
so that's sort of a consequence of our long history with this yeast. I mean, we've been aware of brewing for a long time. So when it came start studying these different organisms, I don't actually know how E. coli became sort of the model bacteria organism, but yeast, the Saccharomyces cerevisiae, became the model eukaryotic organism because it's like relatively simple, but still like a very robust organism that we just have known about for a long time and have been using, like I said, things like fermentations to make beer, to make bread for a long time. And so it became very well studied. And because it was so well studied, a lot of tools for modifying it and genetically manipulating it were developed that work pretty well. So that's sort of like why. It's the combination of the fact that it is a good model, it's a good, it's a good organism for engineering and for doing fermentations. And it also happens to be the one that we just have like so much experience and just accumulated knowledge with, that it just makes it like a very logical choice to use. It's not the only one that gets used though. But.
Cool. So is the idea basically we could look at any, maybe there's some limitations you could talk about. We could look at any organism. If we could take a feature from the genetics of a plant, from an animal, and say this organism has developed the genes to produce this compound, whether it's a pigment, whether it's some sort
potential
of
antibiotic.
An antibiotic.
Yeah.
We can take the genes from that other organism, take them out, and put them into yeast, grow a whole bunch of this yeast, and now the yeast is producing that compound independently.
Yeah. Yep.
That is an incredibly powerful tool. And
yeah.
I get so excited by it.
Yeah, I mean, that was one of the major things. Like I said, I don't make a lot of money, but it was very much like, yeah, I'll spend a bunch of years studying this stuff. This stuff is really interesting, really potentially powerful, has a lot of like promise for the problems that we face and the challenges that we face as a society. Those were all things that really wanted me, pushed me to get deeper into it. And then to just finish the thesis story, we figured out as a lab that, when you grow yeast on these non-native carbon sources, they don't realize that they have food. Like, yes, they're actually able to take it in and sort of metabolize it and grow, but they turn on all of these genes as if they're starving. Like, they turn on stress response genes, they turn on DNA repair genes, they start breaking down like the lipids in the cell for food. So our lab's secret sauce is turning on native regulatory structures that yeast turns on when it's growing on something that it knows and loves. So we're basically tricking the yeast into happily growing on something that it did not evolve to grow on, that it does not natively grow on. And then you see that it grows better and uses these resources more efficiently. So again, that would be something that, like, if you're feeding it X percentage, 100 grams or whatever of sugar, instead of 40 grams of sugar going towards what you want, maybe you increase that by a bunch because it's actually just able to more efficiently grow and utilize those carbon sources.
That is so cool. So what's the end result? Are you guys gonna have are you working towards a paper that you want to publish?
Yeah, that's the currency of the realm, is papers in grad school. So my project is to take that, and he developed a system that lets it grow on this sugar xylose, which is like a 5-carbon pentose sugar. And I'm trying to figure out how to apply that strategy to whatever non-native substrate that you would want to grow on. So I'm going to be looking at trying to grow on multiple different, like, renewable, available, cheap potential feedstocks for fermentation processes, and try and figure out, like, how do I change the regulatory structures of yeast to make it so that it happily grows on all of these things, as opposed to just this one substrate that he figured out.
That is super
but it's
cool.
it's like a really smart, I think it's a really smart project. It's a really interesting project. Like, I love it.
That is super cool. Are there things you can do computationally maybe to simulate the work before you actually synthesize DNA, put it in the yeast and test it? Like, how do you prototype an idea?
So this gets into like the field of metabolic engineering and like what the flow is, like how you attack a problem. So, and we've talked a little bit about this. So you'll identify either some molecule compound that you want to bring into metabolism, in our case, or something that you want to make with the byproducts of metabolism.
Okay.
And there are resources like KEGG, which I think I told you about, or maybe even showed you.
Well,
go ahead.
For EverymanBio, there's a compound that I want to use yeast to produce, and there's 4 genes.
Yeah.
The
Yep.
PhDs have figured out the metabolic engineering necessary to generate this compound, and there's 4 genes that do it. What is the process if I want to take those 4 genes and see if I can use that to engineer the yeast to produce that compound?
Yeah, so you're going to take those 4 genes, you're going to put them onto some plasmid, right? And then you're going to transform the cell with this plasmid. And ideally, at that point, if it's expressing everything, you should start to detect that your cell is making some of this product. And usually what happens is you're able to make a little product, and then there's an iterative process of improvement where you're modifying metabolism or the culture media or several different things.
Yeah.
The conditions to try and increase the amount that gets made. And that happens on like the culture side in terms of like the different media that you put in, but it also happens on like internal to the cell, the metabolism side. So like something about, you'll put in this 4-enzyme pathway, and the cell will probably be expressing all of those enzymes, but the problem is enzymes have different activity levels. They have different sort of like affinities for their substrate. So a lot of times you'll have one enzyme that's running way faster than the other ones, and you'll end up building up like the metabolic intermediate after one enzyme. So what people will then do is then start to like screen different expression levels, different combinations of expression levels of the different enzymes, and just see which one does the best. So this is where you start to get into like a little bit more like industrial metabolic engineering. Instead of just getting the compound that you want, now it's time to like really optimize and maximize what you can get. So they'll create these libraries, like they'll take your plasmid that has 4 things, they'll make 1 million versions of that have different expression levels of each enzyme, because it's very hard to tell beforehand like what's going to work out. A lot of times it's best to just literally throw a lot of combinations at a wall and come up with some situation. Maybe it's 96 well plates where a different combination is in each well. Come up with some way of detecting which one's the best, and then just go with that one. And that's a process that has been like greatly aided by the ability to synthesize DNA more cheaply. You can just do these massively parallel experiments.
How do you control the expression level? Because I kind of just thought it was either has the gene or it doesn't, and either has a promoter which turns it on or doesn't.
Right, so there's different strength promoters. So the promoter is what starts the transcription process to make the mRNA that gets translated. Some promoters, not a lot of mRNA gets made from it. Some promoters, tons of mRNA gets made from it. So
just trying a bunch of different variations of promoters will...
Right. So you might think that like you would just want to just blast a cell with as much of each enzyme as possible to get your product. But that's not actually the case usually. Usually you want to balance it so that in the broader context of the cell's metabolism, everything flows nicely through your pathway. And how
do you measure the amount?
There's a couple other things as well.
Yes, I'm sure. That's
more
the
steps
PhD process.
Right.
So I guess my other quick question is like, what tool do you use in the lab to measure the production of the, I guess you'd call it the metabolite that you're looking for? Are we dealing with a bioreactor that has like a sensor in it that is detecting the production of whatever it is that you're trying to produce?
Usually not. Usually you start at a much smaller scale. Like I said, like a 96 well plate where the volume is I think like 400 microliters max volume.
Yeah.
Where you're testing a whole bunch of different combinations. And then once you identify something that seems pretty promising, then you'd start to put it into like smaller scale and ramp up like size of fermentations.
But how do you measure the amount of compound present in those wells?
There are a ton of different ways, and you have to pick the one that makes sense for the application that you're doing. So like something that you could do would be, let's see.
Like chromatography?
Yeah, chromatography. That's like a very major one. Like you figure out like, okay, what chromatography conditions do I need to run that I should detect like a peak on my my LCMS or HPLC of like where this thing is eluting and then quantify it like that. A lot of people will tag, no, that's the enzymes. You can like tag the enzymes and detect them to make sure that like you can see the different levels of enzymes by like detecting them with enzymatic assays. A lot of times you can do activity assays where like you'll take the cells, lyse them open, and then come up with some way of monitoring the reaction that you want and see like which combination of the different expression levels is best able to make that final product. Yeah, it takes a little bit of like thought and planning.
Yeah.
Yeah, you can generally do it. Some things are easier to detect than others and quantify than others.
If I'm going to start a biotech company and I need to hire someone to run my lab to design those experiments, what's the type of person that I'm looking for? Or is it like a chief scientist? Is it like, what's the title and what does the profile of this person look like, do you think?
I don't know much about titles, but I would think that you'd want like a really good indication would be someone who, like in their PhD or in a postdoc, worked specifically on the organism that you're using, or worked trying to make the compound that you're making or something very, very similar, because then you're going to be getting someone with a lot of that like very specific knowledge to that, you know, particular biological system that would maybe let you skip a few steps. That would be ideal. I mean, someone that's like just excellent in lab and excellent, you know, someone who's just an excellent metabolic engineer would be able to go to the literature and come up with a plan on their own, and they'd be good too, and would just be able to work their way through it. I don't want to say you would need someone with a PhD because clearly, you know, Dakota is able to just do it over time. But like, obviously, if someone has a bunch of experience doing exactly what you want them to do, a lot of times that happens in the sciences in a PhD.
Yeah,
definitely can give you a little more confidence that
that makes sense.
that they're going to be able to do it.
Makes a lot of sense. So you're
Did that answer your question?
Yeah, absolutely.
Yeah.
Absolutely. It's very helpful. So you're at the school right now. I think my understanding is you're right outside the lab. Would you be open to giving us a little tour of your lab?
So just to give a quick tour of the lab space, start by highlighting a nice view of Boston in the distance, a bunch of write-up areas, workstations for the graduate students in the labs that are right across from all of those stations. And as we go in, we can see different benches. Each grad student in our lab gets one of these benches, and they all have a set of pipettes, and we have a lot of common reagents that we all maintain and make together, but we also have a lot of personal reagents that get made, like for me, a whole bunch of different carbon sources that I want to grow my yeast on. This is my bench. Here we have a whole bunch of chemicals that you would need to make different media, different experiments, a whole bunch of chemical standards for running things like HPLC or mass spectrometry. Here we have a bunch of incubators, large incubators, small incubators, centrifuge, and a bunch of fridges and freezers for storing plate cultures that we want to keep using and just put them sort of in a cold temperature to be metabolically slowed down, or all the way to -80 for glycerol cultures that you want to store basically indefinitely at a very cold temperature. And in this room, more tools, centrifuges, refrigerators, spectrophotometer, gel box, HPLC, which is maybe the most expensive thing that we have, or maybe a microscope. There's a microscope, a nice microscope in the back here, but I won't show that. And that's it.
Man, it's very cool. Thank you for thank you to Tufts as well. I think you said you had to ask for permission to come and film in here. So thanks to Tufts, and thanks to you as well for giving us a tour of your workspace, and to your colleagues for letting us bother them with you walking around with your laptop.
Of course. I hope that people were able to see a little bit. Maybe if we want, we can come up with a different way to film it. But obviously, if you have any questions about what's there, the type of stuff that's needed, or if any of your viewers do have questions about, like, you know, if they're trying to do XYZ experiment, I could talk about the type of stuff they might need.
Very cool. Very cool, man. Well, I'm going to send you a link to the episode when it's live. If people want to get a hold of you and ask questions about either, you know, becoming a student or just the research that you're doing, what's the best way to get a hold of you?
I think that Twitter probably right now. I don't post a ton, but I check it pretty religiously. I think I sent you my Twitter handle is Sean F. Sullivan.
Okay.
And then email, I don't know if I want to give out a personal email, but
oh, that's cool. I'll link to your Twitter.
Let's see. Yeah, I should get any messages there.
Okay.
Or if, you know, if people want, they could do sean.sullivan@tufts.edu. Sean spelled S-E-A-N. And that'll be an email address that at least I'll be getting rid of at some point. So
yeah,
can't get too swamped.
Well, man, yeah. This was awesome. Thank you so much for the conversation. I learned a ton.
Yeah,
I could go probably pick your brain for hours on especially the yeast engineering. So
round two at some point. Yeah.
Absolutely. Thanks again,
but
man.
No, man, I'm super excited to follow your journey, and I was really excited to, you know, hear. We met on that that Google group, the DIYBio Google group, after you posted on it.
Yeah.
So I love seeing that energy. People want to go after it and learn about this stuff. I think it's really interesting. So good luck. I'll be keeping tabs.
Cool, man. All right. I'll talk to you soon. See you, brother.
Hey, bye.